Hello. Today we're going to talk about digestion and absorption of fuels. So, a couple things just quick. Still got these two little monsters behind me, but they're pretty quiet. If you hear a little clicking around, it's just them. And the supervisor is pretty unconcerned about us. So, what we're going to do is we're going to talk about digestion and absorption. One of the challenges of biochemistry is that we spend a lot of time on we take glucose from the blood into the cell and we do whatever steps, reactions, processes we're studying and doing to make energy for the cell. We do the same thing with a particular fat. We'll do it with an amino acid. But we spend so much time on that and have our focus on that, we kind of forget there is a process or a conversation to be had about how do those molecules go from the point of being in the food that we eat to the point of being in the bloodstream and available for the cell to take them in and make ATP, make and maintain that consistent supply of energy for the cell. So there's really two topics contained in this, and we'll be talking about both of them. So the process of breaking down the food to give us these single molecules is our digestion. So we've got to break the food down, get the molecules. We've also got to take those little molecules that we make and absorb them, get them into the bloodstream to go to the cells and be available for the cells. So there's a few things we need to talk about and a few commonalities you're going to see through all of what we're going to discuss today. We're going to see our foods, our molecules getting lubricated, homogenized with fluids and everything as they travel along the gastrointestinal tract. We're going to see enzymes getting secreted to break big molecules down into smaller ones. And that's really what we're going to be focusing on, or a trend to keep in mind. We're starting out with the big molecules, and we're going to focus more on the molecular level. Your physiology classes and your anatomy classes are going to talk about the structure of the different organs in the GI tract, the layers, what they do, things like that. We're going to talk about the molecular structure of the component of your food and how it breaks down to its smaller units. And again, we're always going to be going big molecule to small molecule as we travel along the GI tract, ultimately absorbing the small molecule. So we'll be focusing on the molecules and how they work and how they get broken down, as typically is done with the biochem portion of this conversation. We'll have electrolytes, we'll have acids, bases, things like that. So again, we'll talk about them as they directly focus on our story. We'll have bile acids and bile salts. Fats are a little bit of a different story because they're fats, they're not water soluble. So we'll have a little bit different, like I said, different story there with the lipids, with the fats. We'll have further breakdown of different molecules here and there in the small intestine. We'll pull it apart a little bit, talking about, of course, what happens where, a little bit about the layers and the cells in those layers and everything. We're going to have different organs performing different functions. When we're looking at our fats, our carbs, and our proteins, some of our organs have a role in processing all of them. Some of our organs don't process all of them. So we'll go through each individual story. I think that's probably the easier way to do it, and tell the story for the carbs, the story for the fats, the story for the proteins. Another commonality we're going to have in these stories is how some of the enzymes work. We have inactive enzymes in a lot of cases that need to be activated. So we'll have these inactive precursors to some of the digestive enzymes, and we call those zymogens. Then we have some kind of indicator, some kind of stimulus, depending on the enzyme, that activates it. It'll convert the inactive form to an active form. So we'll see that over and over as we look at several of our enzymes. Amylase, which we're going to see with our carbohydrates, is an exception. Lipases are an exception. They're just ready to go. The action of our digestive enzymes, and when I say breaking down, I should be saying hydrolyzing. So we looked at hydrolysis and we looked at how we break a molecule using water, and that was our hydrolysis. So when we break the large molecule into a smaller one, that's how we're doing it, even though habitually I tend to say we break it down. Okay, let's start out with carbohydrates. And really, I think the easiest thing to do is just start drawing. We're going to start out with starches. Remember, they're the big molecules, either linear or branched, with a whole bunch of glucoses connected. We might have sucrose, which is glucose and fructose connected, a disaccharide. We might have lactose from our dairy. That's going to be galactose and glucose connected. So let's just start drawing this. It's probably the easiest thing to do. And we'll just go through sequentially and tell our story, look at our story. So we're going to tell the story like you're sitting there eating a sandwich right now. So part of the sandwich, the thing holding your sandwich together, is your bread. So bread has starch in it, and the starch in bread is in two forms. And we've talked about these. We have amylose, and that's a chain, a linear chain of glucose molecules. And we're going to come back and talk about the structure and draw the glucose molecules that are joined, but they're joined by an alpha 1,4 bond. So when it's linear, we're joining by an alpha 1,4 bond. Then we're going to have amylopectin in our bread. That's the branched starch. So again, we're going to have glucoses. And we'll come back and look at the structure, and both these chains continue. When we're talking about the linear part, again, alpha 1,4 bond. This one's branched though. The branch itself, the connection of the branch, is an alpha 1,6 bond. Then we go back to alpha 1,4. And like I said, we'll talk about that, but those bonds make a difference. Different enzymes break the different bonds. So of course, that's kind of the meat and potatoes of biochem. So what we're going to do is you take that sandwich, you chomp off a bite of it, and you have a bite of that sandwich in your mouth. You're going to do two things. One is you're going to physically be breaking down that bite of your sandwich, that bite of bread that contains those two starches. So physical breakdown is chewing it. You start chomping it up. The other thing you're going to do is you produce saliva, and your saliva contains salivary, as in the saliva, alpha amylase. So first enzyme we're going to talk about in the carbohydrate story. So what the alpha amylase does is starts breaking the 1,4 bonds, starts breaking the 1,4 bonds. So we have some of the 1,4 bonds starting to get broken down, and your sandwich kind of getting mushed up as you chew it. Then you send it on its way. So goes through the esophagus, and the next stop is the stomach. So in the stomach, we've got a couple things going on. So once we hit the stomach, we have some shorter polysaccharide chains. We also have some oligosaccharides. And remember the oligo. It's three glucose units up to, depending on which book you're reading, 6, 7, 8, 9, 10 units. So basically, we have shorter chains of glucoses. So in the stomach, we've got a couple of things happening. One is the stomach pH is lower, so that shuts down your amylase. So amylase is done. It's shut down there. So other than that, really not a lot is happening. We still kind of mix everything around. We've got the polysaccharides getting mixed around. Little bit of hydrolysis happening, maybe turning them into littler pieces. But for the most part, the stomach is just sending these off to the small intestine. So the next part of our story is going to take place in the small intestine. So now, in the small intestine, this is where we're going to do most of the absorption, and this is where we're going to do most of our breakdown. So I'll try to draw this. I don't know how well it's going to go, but we'll try it. So let's say we have the small intestine. We have a piece of it. That's a pretty rough looking small intestine. But what we're going to do is, let's say we're going to take a chunk out of the lumen of that small intestine and just kind of blow it up. So we're going to blow it up and we're going to look at the cells, they're called the enterocytes, that we're going to see when we blow that up. So we've got these cells and they're kind of aligning. And they're the lining. And what we have behind those is we have a blood vessel. We have the bloodstream. So what we're trying to do, what our goal is to get these polysaccharides broken down so that they can be absorbed in the small intestine, get either glucoses, individual glucoses or close to it, absorbed in there so that they can go into the bloodstream and go to the individual cells and be converted to energy or be used to make energy. So that's our job here. So a couple of things happen. One is the pancreas releases some alpha amylase. So this is pancreatic amylase. So the pancreas releases amylase. So more amylase, more alpha-1,4 bonds getting broken. So we're going to break the chain down even more. So our goal is we want to keep breaking and we want to end up with these individual glucoses, get them in the bloodstream and get them to the cells where they need to go. So pancreatic alpha amylase, when you see an adjective like that, salivary, pancreatic, whatever, that's telling you where it's found. When we get further into the breakdown within the cells, we'll be saying things like mitochondrial, whatever enzyme, cytosolic, whatever enzyme. Again, it's just saying where it's found, but this time the where is what part of the cell. So we've got all these polysaccharides still hanging out. So we've got some smaller ones. And what we're going to do is we're going to keep breaking them down. So we've got little disaccharides, got some polys, things like that. So there's some different enzymes that play into this now. So one of them is an enzyme called maltase. When I still have two glucoses hooked together, that's a maltose made up of two glucoses. So if I have a maltase, that'll break those down. So our maltase breaks maltose. So it's going in there. Now we've got single glucoses. Then we can also have, when we get broken down to our glucoses, we also have a way of getting them in there. So we've got another molecule to talk about. We've got our glucoses. we want to get them into these enterocytes. We can also have something called a sodium glucose link transporter. We abbreviate that. And what that's doing is it's also getting the glucoses in there. So what's interesting though with this one though is we have some sodiums around. We have our glucose, and the glucose will go in, take two sodiums with it when it goes. So it's two sodiums with each glucose going in. So we do things like that, and we eventually get all of our glucoses in the enterocyte. Now, our next step is we've got to get those glucoses into the bloodstream. The point of the bloodstream is transport to take them wherever they need to go. So we have another part of these enterocytes called our GLUT2 transporter. So what that does is it takes the glucoses then and puts them in the bloodstream. So we get the glucoses then in the bloodstream so that they can go wherever they need to go. Something Dr. Krabbe's going to talk about is something called resistant starch. So we're talking about this like all of our glucoses are going, getting in the bloodstream and going where they need to go. Some starch will not get broken down, so it'll stay hanging around in the small intestine. What will happen to it is it's either, well, it is going to go to the large intestine, and once it gets there, it either will get fermented there or it'll just get passed through and out of the large intestine as waste. So those are the enzymes, and that's the story on how we're going to get our glucose into the bloodstream. Then we'll take the glucose from the bloodstream to whatever cell it's going to, and we'll go through the steps, the reactions, the processes that we're going to be doing next trimester. Okay, so same story. Okay, next one's lipids. And actually, let's do proteins next because it's a more similar story to carbohydrates than is the fats. So next one we'll do is proteins. And remember what a protein is. A protein is a big long chain of amino acids. So if we're going to break down, if we're going to digest proteins, we might be digesting proteins that we ate, or sometimes we'll break down our own proteins that we're using as fuel for whatever reason. So remember our amino acid structure was the amino group, carbon with the side chain, and the carboxylic acid. Then we said we could take two of these and put them together. Joined the carbon to the nitrogen, pulled the water out of the middle, and formed our peptide bond. So we did that, and we went through that whole story. So ultimately, what we have is a chain of amino acids. One end of the chain has that carboxylic acid end, one has the amino end. So that's what we have. That's what we're going to start breaking down. Probably just easier to draw the picture again. Okay, so we'll do our same story. So you're eating a sandwich. So we didn't get into what kind of sandwich it is because we were only worried about the bread. So you've got a ham and roast beef sandwich and all this stuff in there. But you got your ham, you got your roast beef. So what you have in there is protein in your sandwich. So the protein is that chain, that chain of amino acids. and so on. So you put your sandwich in your mouth, you took a big bite of your sandwich and put it in your mouth. So in your mouth, the only thing really happening is chewing. So you're physically breaking it down. Amylase isn't really doing anything to the proteins. So you physically break it down, mush it up, whatever, and get that sandwich through your esophagus and to your stomach. So next up is the stomach, and that's where chemical digestion starts with your proteins. So this is where we're going to get into this one enzyme activating another story. So we said we've got these peptide bonds. And remember we said if you see pep anything, you're going to get your brain on proteins, amino acids, proteins. So we've got peptide bonds. In the stomach, when our protein gets there, we're going to start secreting pepsinogen. So this is where we said we have these inactive enzymes hanging around, and they get a stimulus that causes them to become active enzymes. This is going to be one of those stories. So the pepsinogen gets activated to pepsin. Pepsin then breaks down or hydrolyzes peptide bonds. So we're going to start breaking peptide bonds and breaking down these polypeptide chains. So next question is, well, why does it become activated? What does that? Hydrochloric acid is your activator. So hydrochloric acid activates it being secreted, actually triggers the conversion of your pepsinogen to pepsin so we could start breaking down the peptide bonds. So now we started breaking, but we have these polypeptide chains. So what we've done is we've just made big chains into shorter chains. So we've got these shorter chains now, but they're still chains. Now we're going to go to the small intestine. So we've kind of got a commonality here. Everything's kind of going to a small intestine story. So we're going to go to the small intestine, and we're going to have the same story again. So we've got the small intestine, and we've got these enterocytes lining the small intestine. So the first part of the small intestine is where we're going, and that's actually called the duodenum. So that's where we're going to be breaking down our proteins. So we've got these polypeptide chains, and we've got them in the duodenum. So when we see those, or the cells see those in the duodenum, the pancreas is going to release some stuff. So the pancreas is going to release trypsinogen, it's going to release chymotrypsinogen, and it's going to release pro-carboxy peptidase, I'm sorry. So we've got two enzymes that end in gen. So we'll talk about that in a minute. So these are all coming from the polypeptide goes into first part of the small intestine, and the pancreas releases these. So we've got the same story. We've got the enterocytes. I'm sorry, I can't talk today. We've got the enterocytes, And that's what everything's going to go through to get to the bloodstream. So we still have the same story. We want to get these molecules broken down into little manageable molecules, get them in the bloodstream, and send them where we need them. So we'll draw our enterocytes again. And we've got the bloodstream running behind them. We've got our polypeptide, and we've got these three molecules. So first one is, let's talk about our trypsinogen. So our trypsinogen is activated by enterokinases. So it's going to be activated by enterokinases, which are enzymes on the surface of the enterocytes. So they're hanging out here activating trypsinogen to give you trypsin. So once I get trypsin, I can break peptide bonds, but it does some other stuff too. So it can start breaking my big polypeptide chain down into smaller pieces. So we're always, always, always going to smaller pieces. But this trypsin we just got, kind of a big deal. So we just got that trypsin. So the enterokinase took trypsinogen, activated it, turned it into trypsin. So it does a few things. One thing it does is it makes more trypsin. Trypsin makes trypsin. So trypsin makes trypsin. Trypsin breaks the peptide bond. So we have more trypsin making more or breaking more peptide bonds. The other thing trypsin does is it'll activate chymotrypsinogen to chymotrypsin. More peptide bonds getting broken. So we're breaking and breaking there. So we keep going, keep going. Then the other thing trypsinogen, or trypsin, I'm sorry, does is it activates pro-carboxypeptidase to carboxypeptidase. Oh, that should have had a new color. This is going to break the polypeptide chain, but break it from the carboxyl end. So remember with our chain of amino acids, we have the amino end, we have the carboxy end. This one's breaking from the carboxy end. So we're breaking and breaking and breaking until we get down to now we've got some small polypeptides. We have some dipeptides. And we still have some tripeptides. So we still need to break down further. We're not done yet. So we've got a couple more or one more enzyme really in the story on how we do that or what we're going to do. So the next enzyme we're going to have in the story, we need more room for this. So we'll just draw one enterocyte. And we have the polypeptides, we have the dis, and we have the tris. So the next enzyme we're going to talk about in this story is we have what's called brush border enzymes. They're kind of hanging out on the top of that, the top edge of the intestinal cell. So that's our brush border enzyme. And there's actually multiple ones. We're going to stay a little general with them, but we're going to talk about two of them. We're going to talk about a dipeptidase, and we're going to talk about an aminopeptidase that breaks down from the amino end. So what they're going to do is the dipeptidases, they're going to take they're they're going to take the dipeptides into the cell. So dipeptidases will take the dipeptides in. They're going to go into the cell by a co-transporter with a hydrogen and get hydrolyzed by the peptidases to get an amino acid. So they can go from there. Don't forget we have the bloodstream down here. Those amino acids then can go by diffusion into the bloodstream. So same as the tri, we could have the tripeptidases. So same story. Well, I guess my arrow was going more that way. They're going to go into the cell through a hydrogen co-transporter as well. Go down to amino acids and go in. So we're going to have a hydrogen co-transporter. The hydrogen will get pumped back out in exchange for sodium. So that's a little more of a story. So then we've got the individual amino acids. They're going to go in. Their individual amino acids then can go in through a channel with the sodium. So we'll do another enterocyte. Need more room. So we still have the individual amino acids that need to go in. That individual amino acid with the sodium, that's going to go in. So, and again, get the amino acid in there and it'll go into the bloodstream. So we end up in all of this with a net absorption of sodium, and this will lead into another topic. Because we're absorbing the sodium, we're going to absorb water with it. Then we'll reabsorb the sodium, get into this whole sodium-potassium pump story. So we'll do that in a different topic. So that's our amino acids getting to the bloodstream. So now the last thing we've got to get to the bloodstream, let's go back, and we've got to do fats. So fats are going to be a little bit of a different story. So we need to talk about the structure a little bit with our fats. So most of our dietary fat intake is free fatty acids. We've talked about those. That was that chain of carbons. We have all our hydrogen, however long with the carboxylic acids on the end. Some of our dietary fat intake is triglycerides or triacylglycerols. What those are is a molecule of glycerol, 3-carbon chain, and 3 fatty acids joined to it. And when we're making them, we'll come back and talk about the structure and everything, the bond, the ester bond that connects the fatty acids. Cholesterol, we're going to do it another story. Phospholipids just have the phosphate in there in between that 3rd carbon and the fatty acid. Fat soluble vitamins we'll mention, but we'll do in another story too. Okay, again, let's just draw pictures and do this one. Okay, so we'll do the same thing. Most of the fats that you eat, so we'll say, okay, you're eating your sandwich. So we've got the sandwich and we have the bread and we have the ham and I don't know, we said roast beef in there. Let's say we threw in some mayonnaise or some butter or some margarine, whatever. So what we're going to get in that mayonnaise, the butter, the margarine, whichever, is we're going to get triglycerides. So a triglyceride is a glycerol backbone, and we'll talk about that word. So a molecule of glycerol, you can throw the carbons and stuff on there, and we'll talk about these bonds. It's an ester bond. but it's three fatty acids attached to that glycerol backbone. And that's a lot of the fat that you take in. So we're going to start breaking them down in the mouth. And you're going to do like we have before, two things in the mouth. You're going to chew your sandwich. That's a physical breakdown. You're also going to secrete lingual lipase. Remember we said lip anything, you're going to get your brain on fats. That's going to start the fat breakdown. So we'll start in the mouth, start the fat breakdown there, and we'll ship everything off to the stomach. Maybe. Okay, got here. So when we get to the stomach, we have gastric lipase. So your enzyme, first part of the name is describing the location. We have a gastric lipase. What that's going to do is it's going to keep breaking down. So we still have the triglyceride or triacylglycerol chunks really, and they're going to go now to the duodenum. So a little more breakdown, not a lot going on, but now we're going to go to the duodenum. When we get there, we've got a couple things. We've still got triglycerides or triacylglycerols, abbreviated as a TAG, and we've got some fat droplets. This is where the fat story starts deviating a little bit from being similar to the carb and protein story. So when we go to the duodenum, remember we have the acidity in the stomach. We're going to take that acidity with us at first. So we're going to take that acidity with us. So now we've got to do something there. So we're getting to the small intestine now. We're in the duodenum, and we've got two hormones that are going to get secreted, plus some other stuff. But we're going to stimulate the small intestine cells to make these two hormones. One is cholecystokinin. Other one is secretin. So we're stimulating the small intestine to release these. So cholecystokinin, we abbreviate it CCK, and it's got a couple jobs. What it does is a couple things. It stimulates bile production. It also stimulates the release of pancreatic enzymes. Oops. So now we have pancreatic lipase in the story, breaking down our fats even more. So what the bile does, or specifically the bile salts, is they help with emulsification. Because remember, we have fats, they're not water soluble. So basically what we're doing is we're coating the lipid, allowing the pancreatic lipases to do their thing. So what we're going to do is we're going to break the fat down further into fatty acids and monoacylglycerols. So the monoacylglycerol is still the glycerol backbone, but only one fatty acid attached to it. Then we have the secretin. What it's doing is it's releasing bicarb. Because remember what we said, we still have the acidity from the stomach. So we've got to release bicarb and get the pH back up to about 6.5. We've got to get rid of that acidic environment. So now what we've got is we've got the fatty acids and we've got monoacylglycerols hanging around. So this is what we've got to deal with Now, so what we're going to do is we're going to have a micelle. So a micelle is kind of this ball of broken down fats, bile salts, cholesterol, all coated with phospholipids. So just kind of a whole bunch of fats mushed together. So by having all of this mushed together, that micelle then is a form in which we can transport those monoacylglycerols and the fatty acids across the enterocyte. So we've moved on a little further down in the intestine, middle of the small intestine, and we're going to pass this whole thing through the enterocyte. So same thing, we got our little enterocyte. The bile salts don't get absorbed here. They're going to go to the ileum, go back into the portal circulation, but our monoacylglycerols and our fats do go in. So they're going to get absorbed, but they're going to get packed into what's called chylomicrons. So something else we're going to talk about, and they're going to bind to the basal membrane. And they're going to get transported. This is where we get a little bit different into the intestinal lymphatics. So they're not going directly into the bloodstream like we did with the carbs and with the proteins. So then from the intestinal lymphatics, then they can go into general circulation. So a little bit of a side trip there, a little bit of a catch glitch in the story. So that's our story with getting our fats then into the general circulation. Then they can go to the cells through the bloodstream and do the steps, the processes, and everything that we'll be doing in class. Something else, just a little bit of a side note we'll be talking about, we'll come back, is fat-soluble vitamins. They need to go through a very similar story to this. Your vitamins A, D, K, and E, where your Bs and your Cs are water-soluble.